Welcome to my presentation titled Ce3D Enhanced Tissue Clearing for Multicolor Microscopy. In this presentation, I will start by providing a background on tissue clearing, followed by introducing Ce3D, our tissue clearing reagent that we have recently launched. This introduction will be then followed by current BioLegend product offerings for tissue clearing, relevant web pages, and references. And lastly, I will provide a brief summary of what I have covered in this presentation. So let's start with a background on tissue clearing. Tissue clearing is a process that renders tissue transparent, enabling researchers to look at biology in 3 dimensions. Light scattering is one reason why tissues appear opaque. This is due to biological tissues being composed of heterogeneous components such as water, lipids, and proteins. Each of these components has a different refractive index, or RI, and as a result, they interact differently with light passing through the tissue. This causes light scattering in a tissue and tissue non-transparency. Light absorption is another reason why tissues look opaque. This is due to endogenous light-absorbing pigments such as hemoglobin, which reduce light propagation, therefore contributing to low tissue transparency. As you can see in this slide, tissue clearing reagents and methods aim to make tissues transparent by reducing light scattering in tissues, by normalizing the RI throughout the tissue, and by minimizing light absorption by discoloring the tissue. Tissue clearing offers many advantages, some of which are listed in this slide. For example, tissue clearing can help understand the spatial composition, distribution, and function of different cell types. This information could be useful for determining cell positioning, relationship between cells and their microenvironment, and phenotypic and subtype identity. Tissue clearing can also help us gain better understanding of biological processes such as functional states, signaling events, and cell-to-cell interactions. In addition, tissue clearing may be useful in translational or personalized medicine. For example, it may enable scientists to determine the morphological and spatial alterations and interactions of subset of cells in tissues from disease versus healthy individuals, and tailoring treatments based on these observations. BioLegend has recently launched a tissue clearing reagent that is called Ce3D, which stands for Clearing Enhanced 3D. So what is Ce3D tissue clearing reagent? Ce3D stands for Clearing Enhanced 3D. It is a hydrophilic aqueous refractive index matching tissue clearing reagent. It has an RI of about 1.5. Ce3D allows easy, rapid, and robust tissue clearing that is compatible with fluorescence microscopy, meaning that it preserves fluorescence signal from diverse reporter proteins such as GFP and organic fluorophores such as Alexa fluorophores. The Ce3D tissue clearing method works by simple immersion and does not require special equipment. Therefore, it is a simple one-step passive clearing process for rapid tissue clearing. Using this method, lipids, cell morphology, and tissue architecture are preserved. In addition, this clearing reagent works on tissues that were fixed with 1 or 4% PFA. Tissue clearing with Ce3D is reversible to allow 2D IHC. An added benefit is that we've validated over 60 conjugated formats using this reagent, and provide a list for you to select antibodies from to simplify your multicolor microscopy options for tissue clearing. In this slide, I'm showing the Ce3D tissue clearing workflow, which is about 7 to 8 days. This workflow covers time from transcardial perfusion and fixation to when the tissue is ready to be mounted on slides and imaged. There are similarities between steps involved in 3D compared to 2D IHC. Both 3D and 2D IHC require permeabilization, blocking, and antibody labeling. Antigen retrieval is an optional step depending on the antibody and target used. The major difference between 3D and 2D IHC is that 2D IHC is limited in spatial and morphological analysis of tissue microenvironment due to the thinness of tissue sections. 3D IHC and tissue clearing, however, allow analysis of larger tissue sections greater than 100 microns and organs. In this slide, I'm showing you examples of 500 micron tissue sections from liver, spleen, and intestine that were harvested from a mouse that was perfused with 1% PFA. As observed, if there is a need for tissue histology, such as H&E staining, tissue clearing can be reversed by simply immersing the cleared tissue in PBS for 1 hour. In this slide, I'm showing 3D IHC videos of 500 micron spleen and intestine tissue sections from a mouse that was perfused and post-fixed with 4% PFA. In this video, to the left, you are observing the tissue section from mouse intestine that was stained with antibodies against CD326 or EpCAM, CD45, and tubulin. CD326 is a marker that stains intestinal epithelial cell linings, the microvilli. CD45 is a leukocyte marker, and tubulin stains the innervating neurons. In the video to the right, I'm showing tissue section or a video from tissue section from a mouse spleen that was stained with CD3 epsilon, CD45R, and DAPI. CD3 epsilon is a T cell marker, CD45R is a marker for B cells, and DAPI stains the nuclei. The video demonstrates the spatial localization of B and T cells within B cell and T cell zones of a mouse spleen, respectively. The antibodies used in these videos are available at BioLegend. In this slide, I'm showing 3D IHC videos of tissues from mouse, lung, and brain. These videos have been kindly provided by Dr. Ronald Germain from the National Institute of Allergy and Infectious Diseases at NIH. The tissues shown here were cleared with Ce3D tissue clearing reagent. Here, I want to draw your attention to the fact that tissue clearing with Ce3D preserves the fluorescent signal from various reported proteins. In the video, in the left panel, you're seeing the lung tissue section from a CD11C YFP transgenic mouse that was stained with anti-cytokeratin antibody. The 3D video nicely demonstrates the association between dendritic cells and the bronchial epithelium. In the video to the right side, I'm showing a 1 millimeter brain tissue section from a confetti mouse brain, which demonstrate individual neuronal cell bodies and axonal processes in the hippocampal and isocortical regions of the cerebral cortex. The confetti reporter mice express GFP, RFP, YFP, and CFP in distinct cells. So here you're looking at YFP and RFP signals in neurons. So in the next section, I'm covering BioLegend Ce3D product offerings, web pages, and references. Ce3D tissue clearing reagents are available in 2 kit sizes. Each kit includes the Ce3D tissue clearing solution, the permeabilization and blocking buffer, the antibody diluent buffer, and the wash buffer. These are the reagents that are needed to carry out the tissue staining and the tissue clearing process. Individual kit components are also available as standalone reagents and are offered in 2 sizes. You can visit our website to learn more about Ce3D relevant information, including the reagent itself, a complete protocol for the Ce3D workflow, troubleshooting guidelines, as well as a list of BioLegend validated antibodies for use with Ce3D. You can also access references to learn more about the utility of this product. The next section provides a summary of information provided on Ce3D. In summary, Ce3D is a hydrophilic tissue clearing reagent with an RI of 1.5. Ce3D allows a simple one-step passive clearing process for rapid tissue clearing. The reagent is compatible with multicolor fluorescence microscopy and is reversible to allow 2D IHC. Ce3D reagents have been validated with a number of BioLegend conjugated antibodies and are available in 2 kit sizes and as standalone reagents. We thank Dr. Ronald Germain at the Lymphocyte Biology Section, Laboratory of Systems Biology at the National Institute of Allergy and Infectious Diseases at NIH for developing the Ce3D tissue clearing reagent. Thank you for attending my presentation. For more information, visit biolegend.com/Ce3D.